Great. Well, I think that we're ready to get started. Thank you all for joining us today in the Dana Farber Connect to Science seminar series. Before I introduce our speaker today, I wanted to give you a couple of quick reminders, including that if you have a question during the course of the talk that you want to communicate to Chris, you can do that by filling out the question in the Q&A section at the bottom of your screen. Also, if you take a moment to visit the website that's linked from Chris's title page here, you'll be able to catch up on the future Connect to Science speakers that we've got lined up in the coming months as this series continues. So I'm really excited to introduce Chris today. Not only is he an excellent scientist, but a good friend of mine. So it's a particular pleasure to have him here with us. Chris is no stranger to Boston, given that he was actually awarded his PhD in chemical engineering, originally from MIT, and then stayed on to work with David Sabatini for his postdoctoral studies at the Broad Institute of Harvard and MIT. He, in 2012, Dr. Wood, I should say, launched his independent laboratory at Duke University and has been incredibly successful in establishing an exciting line of research focused on cancer therapy resistance. And that's part of what we're going to hear about today. Chris has received so many awards for his exciting research, including recently being awarded by the Department of Defense with the Breakthrough Award, previously receiving the V Scholar Award and the Liz Tilberis Early Career Award from the Ovarian Cancer Research Fund. Chris, I should also mention that Chris received the prestigious Forbeck Scholar Award back in 2012 and has been recognized numerous times for his excellent mentorship. We're so glad to have you here, Chris, and to hear the latest about your research. So take it away.
Okay, well, Chris, thank you so much. Those of Chris's of us in the world who spell our names in a strange way have to look out for each other, and I appreciate the invitation to be here. I do also just want to say that a special thank you to the folks who have organized and put on this Connect to Science series, not just for the invitation to be here today, but really for what I think has been a real service to the cancer research community during a difficult and trying time. And I know a lot of colleagues really all over the world that are tuning in to these seminars. And so thank you for that service. I'm happy to be here today and talk about work that we've been doing focusing on how the BCL2 network that regulates mitochondrial apoptosis evolves and changes during tumorigenesis and therapy-related tumor evolution. And it's worth saying that, you know, a lot of the work that we do related to the BCL2 network actually stems in some ways to my meeting Chris Soroshik and through him, Tony Latai, at a Forbeck meeting about 7 years ago. And that really got us thinking a lot about apoptotic regulation in cancer and has really been a source of a lot of inspiration for us. So we're always grateful to them for that. Okay, so why mitochondrial cell death? I think that one of the things that many in this audience will appreciate is the fact that mitochondria are well known as entities in the cell that can sense environmental changes and oncogenic pathways, and they transduce those that sensing into regulatory changes to the BCL2 network, the network of proteins that work together in a sort of stoichiometric tug of war to determine ultimately whether a cell commits to apoptotic cell death via the release of cytochrome C into the cytoplasm, a process that is not only important for cancer, but that also we increasingly appreciate that it regulates other processes aside from cell death, like inflammation. And so our interests in this space have really been motivated by the fact that the mitochondria, through their regulation of the BCL2 network, have the potential to exert broad control over cell survival and immune surveillance in cancer. And as such, if we could better understand really how those decisions are made by the mitochondria and how these regulatory steps work, we might be able to derive therapeutic strategies with selective anti-cancer activity. So, you know, I mentioned that we know that mitochondrial apoptosis the decision to undergo mitochondrial apoptosis is dictated by the opposing functions of the pro and anti-apoptotic BCL2 family proteins, and ultimately the way that those proteins regulate mitochondrial outer membrane permeabilization to drive caspase-dependent cell death. And this network of proteins and their functions in the cell have been worked out over the course of the last couple of decades by really exquisite and beautiful biochemical and cell biological studies performed by a number of labs. Our work has been motivated not necessarily to try to add to this really beautiful body of work, but rather to ask how more peripheral processes that exist in the cancer cell impact the regulation of this network. Those include things like altered oncogenic signaling pathways, metabolic pathways, changes to the physical structure of mitochondria through fission-fusion dynamics, and even the treatment history or evolutionary history of a tumor, how all of these things more that are kind of separate from this network are regulating this network and the decisions that come from it. And our sense, our hope is that by understanding these things, we'll be better able to design therapeutic strategies like direct BCL2 family inhibitors or extrinsic pathway agonists that can modulate this network. And so our contribution, I think, in this space has generally been to understand how these processes that are more distal to the direct protein-protein interactions and that dictate MOMP, how these processes regulate the mitochondrial apoptosis network. So one of the questions that we asked when we first started our work was, are tumors really apoptotically competent? How often are they apoptotically competent? And what BCL2 family proteins do they depend upon in order to prevent themselves from committing to an apoptotic cell fate? A postdoc who joined my lab a number of years ago, Ryan Soderquist, took on this question and used, I think, a very simple but really an elegant and powerful approach to try to dissect this question. What he did was he took a large panel of cancer cell lines. You're looking at something like 60 or 70 of them in this heat map, but he ultimately did studies in something like 100 different cancer cell lines, highly annotated cell lines. And what he did is he took these cell lines, each one of which is represented by a row in this heat map, and he treated each cancer cell line with either a selective BCL2 inhibitor, a selective BCL-XL inhibitor, a selective MCL1 inhibitor, or all possible 2 and 3 body combinations of those drugs. And when he did that, treating at doses that are known to selectively and largely completely inhibit the drug's intended target, he measured cell death. And in so doing, he was able to infer the particular BCL2 family proteins that are restraining cell death as a function of the particular tissue and oncogenic mutations present in that cancer. At a very high level, what Ryan learned from this study were a few things. First, there are a subset of cancers that are highly dependent upon BCL-XL or MCL1 for their survival. I'll note parenthetically that these were largely solid tumor cell lines, so we didn't see a lot of BCL2 dependent lines. We would have, of course, seen that had we looked at blood cancer cell lines more extensively. And interestingly, he also observed that there's a very large proportion of solid tumor cell lines that are not necessarily sensitive to the inhibition of BCL-XL alone or MCL1 alone, but that are highly sensitive to the combined inhibition of BCL-XL and MCL1. He also learned that there are a subset of cancer cell lines that don't respond to any of these 3 BCL2 family antagonists. But interestingly, we learned that these cell lines, far from being apoptotically incompetent, are rather simply dependent upon other less well-studied members of the BCL2 family like BFL1 and BCLW. So this study gave us, I think, a little bit of a global view and really pointed to the idea that the mitochondrial apoptosis network is functional and competent in the great majority of cell line models that we work with. And so the next question that we asked in our studies were, how do oncogenic signaling pathways regulate the BCL2 network. And this work happened kind of contemporaneously with a lot of studies in other laboratories, many of which have ultimately arrived at a similar conclusion. The answer to the question of how is the apoptotic pathway regulated by oncogenic and other signaling pathways in cancer cells is that driver oncogenes exert really disproportionate control over this network. And the work in our lab that really elucidated a lot of this was done by Grace Anderson and Peter Winner. And I'll just give you one example of that. And that's work that we did in the setting of PI3 kinase mutant breast cancers. So in this particular setting, these are PI3 kinase driven breast cancers. What we found was that the signaling through the PI3K AKT mTOR pathway through its control over cap-dependent translation exerts important control over the expression of the MCL1 protein. Now, PI3 kinase mutant breast cancers, like a lot of other solid tumors, are codependent upon both MCL1 and also BCL-XL. And so inhibiting the PI3 kinase pathway in these tumors is sufficient to destabilize MCL1, but not to get rid of the apoptosis inhibitory functions of BCL-XL. This work predicted that a BCL-XL should cooperate with inhibitors of this pathway to drive synergistic apoptotic cell death. And in fact, that's what we found. So if you look at PI3 kinase mutant cell lines versus wild type cell lines, we find a high degree of synergistic cell death induction when you combine, for example, an ATP competitive mTORC1/2 inhibitor with the BCL-XL inhibitor. And these results translate, of course, into animal models and are now actually the basis for the ongoing design of a human clinical trial. Another question that we asked in the group was, how do metabolic pathways control the BCL2 network? It, of course, stands to reason that in order to maintain homeostasis, cancer cells and other cells need to have crosstalk between metabolic pathways and the apoptotic network, pathways that are oftentimes co-localized in and around the mitochondria. But the mechanisms by which this crosstalk occur are not particularly well understood. And so two students in the lab, Kevin Lin, an MD PhD student, and Abby Shi, who's at the time was a Duke undergrad who spent 5 years in my lab and is now an MD PhD student in New York, conceived of, I think, an elegant approach to deciphering the crosstalk between metabolic pathways and the BCL2 network. What they did is they took an AML cell lines, which are highly BCL2 primed, and they treated them with low doses of a selective BCL2 inhibitor, venetoclax, such that they brought those cells right to the edge of the apoptotic threshold. And then they used the CRISPR-Cas9 system to systematically mutate all metabolic enzymes and transporters. And they asked which of those genes, when knocked out, make the cell more or less primed for BCL2 dependent apoptosis. What they found, interestingly, in their analysis were sets of genes that were largely clustered in defined metabolic pathways. And the map that you see here is an attempt to provide kind of a bird's eye view of some of the key findings that we had. Some of the pathways, like nucleotide synthesis pathway that we implicated in our analysis, were not at all surprising. In fact, had been revealed indirectly in prior studies. But one pathway that did strike, that really did catch our attention and our interest was the heme biosynthetic pathway, a pathway that seemed to strongly potentiate the effects of BCL2 inhibition when inhibited. Ultimately, through a series of careful mechanistic studies, what Kevin and Abby were able to show was that heme, which is incorporated into a large number of proteins in the proteome, is important for apoptotic commitment because of its incorporation in the electron transport chain, particularly in complex 4. And as a consequence of this, when heme is depleted, complex 4 is destabilized, and this potentiates mitochondrial outer membrane permeabilization in the presence of a BCL2 inhibitor. Now, this work was done in the setting of AML, and so a couple of other interesting things were observed that are particularly relevant to that disease. First, we learned that the apoptotic map of the cellular metabolism that we generated in this study is really fully decoupled or uncoupled from genes that are simply important for essentiality. So the genes that sensitize to BCL2 dependent apoptosis are non-overlapping with the genes that are just generally essential. So these screens really were pointing to specific interactions with BCL2. Heme biosynthesis, as I mentioned, was observed to play an unexpected role through its regulation of complex 4 and the ETC. And finally, particularly relevant to AML, we found that changes in heme synthesis that occur during disease progression are likely to actually be fueling clinically observed sensitivity and resistance patterns to apoptosis-inducing therapies. Another aspect of cell physiology that we were interested in with respect to how it talks to the apoptotic network are mitochondrial form and function, particularly as controlled by mitochondrial dynamics. So one of the things that folks can, I'm sure, broadly appreciate is that the mitochondria are not static organelles. Rather, they undergo reversible cycles of fission and fusion mediated by a well-described and defined set of proteins that regulate fission and fusion cycles. Historically, folks have thought a lot about fragmented mitochondria as being conducive to increased rates of proliferation and glycolysis, whereas fragmented mitochondria are conducive to decreased levels of proliferation and increased oxidative phosphorylation, although the degree to which these trends hold true across cancers is not clear. A wonderful student in my lab, Grace Anderson, who I already mentioned in a previous slide, became interested in how mitochondrial fission fusion dynamics might regulate not only the apoptotic state of the cell, but really the state of a cell's therapeutic sensitivities. And so the first thing that Grace did was to ask the question whether mitochondrial dynamics are likely to be robustly altered in human cancers. And the way that she did that was to take a genetic approach. She went to the TCGA and she looked at different cancer types and she looked for recurrent alterations in the genes that canonically regulate mitochondrial fission fusion cycles. And one of the things that she observed was that a number of different solid tumors are characterized by recurrent amplifications in the genes that regulate fission fusion cycles, suggesting that genetically hardwired alterations to mitochondrial dynamics are found in a substantial fraction of human cancers. The next thing that she did was she asked whether altered mitochondrial dynamics, whether they are the result of genetically hardwired changes to mitochondrial dynamics proteins or other signaling abnormalities in cancer cells, whether mitochondrial dynamics impacts drug sensitivity. And so the way that she did this is she constructed isogenic models of cancer cells where she forced them into mitochondrial fragmented or fused mitochondrial states by modulating the proteins OPA1 and encoded by the proteins encoded by the genes OPA1 and DNM1L, which is also known as DRP1. What she found in this analysis was that if she forced cancer cells to have either constitutively fragmented or constitutively fused mitochondria, she could potentiate sensitivity to certain drugs. One of the most interesting findings from this effort, which was an unbiased drug screening effort, was that one particular class of drugs, those drugs that impinge on the mitochondrial apoptosis pathway, that the toxicity of these drugs is potentiated when mitochondria are forced into fragmented or fused states, which was an interesting observation because many drugs' activities were potentiated when mitochondria were forced into, for example, a fragmented state, but not a fused state, or vice versa. But with these drugs, we saw this effect in both directions. BV6, for example, is an IAP inhibitor, a SMAC mimetic, whose toxicity is potentiated by genetic manipulations that force constitutively fragmented or constitutively fused mitochondria. And you can see that here. You can also predict or identify cell lines with naturally increased sensitivity to SMAC mimetics simply by identifying those tumors that have amplifications in these canonical mitochondrial fission fusion genes. So for example, if you take lung cancers and you separate those that have amplified canonical fission fusion genes versus those that don't, we find those with amplifications have hypersensitivity to the SMAC mimetic. Ultimately, a series of careful mechanistic studies by Grace and colleagues taught us that, in fact, this sensitivity to IAP inhibitors in cells with altered mitochondrial dynamics do not occur through, this represent a convergent phenotype driven by different mechanisms. So in cells in which DRP1 has been knocked out and mitochondria are constitutively fused, we find that passive leakage of cytochrome C is what potentiates IAP inhibitor toxicity in the cytoplasm. By contrast, in cells in which OPA1 is knocked out and mitochondria are constitutively fragmented, what we find is it's the activation of an ER stress pathway characterized by ATF4 and CHOP that sensitizes these cells to IAP inhibition. So the summary of this body of work was that mitochondrial dynamics and alterations in mitochondrial dynamics are yet another aberrant feature of cancer cells that has the potential to regulate the BCL2 family network and to be exploited for therapy. Mitochondrial morphology genes are recurrently amplified in human cancers. Their amplifications or other genomic alterations are associated with changes in drug sensitivities, and tumors with altered mitochondrial morphology can be selectively targeted. And then the last example that I want to give you of our sort of high-level work to identify ways that the BCL2 network is regulated in cancer focused on cancer treatment history. And this has really been an area that we've thought a lot about in the lab in recent years. So our lab has been interested in using sort of diverse technological strategies to understand aspects of tumor evolution and therapeutic resistance. So how is it that tumors evolve in the face of therapy? Now, to do this work, we have developed technologies like pathway activation screening, methodologies that rely on computational inference from genomic data sets, and we've adapted CRISPR-based methods to do chemical genomic screens, all in the service of answering really a fundamental question, and that is not necessarily what are the mechanisms that drive resistance to therapy, although we do find that question interesting, but really what we're interested in is a question that is perhaps one level above that question, and that is, are there convergent features of different resistance paradigms? And the real motivation for us here is that we know that resistance can be caused by lots of different types of mutations and other alterations in cancer cells. And we know that in the bodies of individual cancer patients with advanced disease, that oftentimes many different resistance mechanisms are operating. And so rather than trying to define and target individual resistance mechanisms, we've been very interested in the question of whether resistance mechanisms might converge biochemically or in functional ways that would allow us to target them in a more effective way. This body of work led to a set of interesting findings in two different cancer settings. One is in AML, and the other is in melanoma. And what we found in these two settings is that if you take AML cells and you evolve them to resistance to BRD4 inhibitors, what you find is that the cells that eventually develop acquired resistance to BRD4 inhibition, which many of you know works at least in part through its suppression of MYC, is that these resistant cells that come out ultimately have found ways of overcoming MYC suppression. And so they have hyperactivated MYC activity in the setting of resistance. Interestingly, a completely separate study in our group in melanoma, focusing on mechanisms of resistance to BRAF inhibition, revealed something very similar, and that was that in very diverse models of resistance, driven by many different upstream mechanisms, that a common and convergent feature of resistant cells is that they also upregulate MYC during the development of resistance. So MYC is downregulated by MAP kinase inhibitors and is reactivated in the setting of resistance despite the continued presence of MAP kinase inhibition. And we see that in lots of cell lines. So this is one cell line, another, and so on. This, by the way, the AML work is the work of Kevin Lin and Justine Rutter. The melanoma work is the work of Katie Singleton and Lauren Crawford. So at a high level, in some, I think what we observed is that in both AML and melanoma, that although there are many different distinct genetic mechanisms that can lead to resistance to these important therapies, um, and although these different mechanisms drive resistance through different effector pathways, ultimately these pathways converge to activate MYC such that MYC becomes necessary and sufficient for diverse resistance mechanisms. And it's ultimately the activation of MYC that drives resistance in both of these settings. Now, that was really striking to us because it implies that rather than thinking of resistance as a heterogeneous set of states that would be very difficult to control, we might actually be able to think of tumors as existing really in only two states. These tumors could exist in either a BRAF-driven state if it's a melanoma, or a BRD4-driven state if it's an AML, and in that state, they would be therapy sensitive, or they can exist in a MYC-driven state that is therapy resistant. And so that observation suggested to us that if we could identify drugs with particular activity in the setting of MYC-driven resistance, that we might be able to construct two-body combination therapies that have the unique property of selecting against resistance. Because you could imagine that in the melanoma setting, you could combine a BRAF inhibitor, which effectively targets sensitive cells, with a drug that exploits the MYC-activated state to specifically kill resistant cells. And if those two agents are combined, then it might be very difficult for a tumor to ever develop resistance. Similarly, that pre-treatment with the BRAF inhibitor ought to select for cells that are hypersensitive to drugs that exploit MYC hyperactivation. And it's here where our work really circles back to our thinking about apoptosis, because one of the things that has been known about MYC for a long time is that MYC hyperactivation in cancer cells can drive apoptotic priming. That is, it can bring cells closer to the threshold of apoptosis. And so we wanted to dig into this in the setting of AML. The thing that we postulated, both from prior knowledge and also as borne out in some of the CRISPR screens we had done, is that MYC hyperactivation, this common feature of BRD4 inhibitor resistant AML cells, ought to predispose these resistant cells to sensitivity to BCL2 inhibition because of the role of MYC in promoting apoptotic priming. We went into a number of different diverse cell line models, and we proved that that was the case. So if you take cell lines and you evolve them to resistance to JQ1, that is associated with hypersensitivity to ABT199, the BCL2 inhibitor, also known as venetoclax. We see similar results in vivo. So if you take a PDX model of AML and you treat the cells from this model either before they've ever been exposed to therapy or after they've relapsed following therapy with a bromodomain inhibitor, what you find is that the relapsed tumor cells have significantly enhanced sensitivity to BCL2 inhibition, again, associated with this MYC hyperactivation. This suggests that the order in which these drugs are applied matters. That is, that if you could first treat an AML with a bromodomain inhibitor, then what you would effectively be doing is selecting for cells that have MYC hyperactivation and that, as a direct consequence, are hypersensitive to BCL2 inhibition. We tested that in PDX models, and I'm just going to give you one example here, where we showed that indeed, if you measure the leukemia burden in the bone marrow, what you find is that if you treat cells continuously with JQ1 or 199, you find a certain degree of disease burden. If you treat with 199 and then switch to JQ1, you don't really affect that. But if you treat first with JQ1, effectively selecting for the 199 resistant cells, and then treat with 199, what you can achieve is a substantial eradication of leukemia cells in the bone marrow. And there's evidence from CD13, CD33 staining that indeed this is associated with apoptosis rather than a differentiation phenotype. And so We have also done a fair amount of work looking at extrinsic pathway agonists, but just for the sake of brevity, I'm not going to go into that today. So at a high level, I think in this just first kind of high level overview of some of the work the lab does, what I would hope to just convey to you is that we have tried to build on the extraordinary work that was done in the decade or two that preceded our work, which really detailed in beautiful biochemical and cell biological detail how apoptotic commitment is regulated in the mitochondria. And we tried to build on that understanding by trying to connect that regulatory apparatus with things like altered signaling pathways, metabolic pathways, mitochondrial form and function, and even the effects of treatment history, to basically more clearly discern how the properties of cancer cells, the unique stresses that cancer cells undergo, might position their mitochondrial BCL2 network for effective targeting. Now, in the remaining portion of the talk, I'm going to tell you one specific story that's, again, related to apoptotic commitment during tumor evolution, but from a slightly different perspective. So one of the things that we appreciate is that many treatment paradigms involve the diagnosis of a tumor in a patient, the application of a therapy that eradicates many of the cells in that tumor, but leaves behind some minimal residual set of cells, cells which are really important because since we can't eradicate these with the initial therapy, they can serve as a reservoir from which eventual resistant progression will emerge. And so this general trend of a tumor burden at diagnosis, a reduction in tumor burden following initial therapy, and then the eventual expansion and diversification of resistant clones is a common pattern that's observed in many treatment paradigms in cancer. One of the things that we appreciate, for example, as I described in my previous slides, is that once tumors become fully resistant, they can become quite heterogeneous and as a consequence, very difficult to treat. And so in this study, we've been focusing our efforts mainly on this residual tumor cells, those that survive upfront treatment but have not yet expanded and diversified into what could be a more difficult to treat, frankly resistant tumor. So this work, which was really led by a wonderful MD PhD student in the lab named Moaz Ali, really started with, I think, a bit of a random observation in the lab. What Moaz observed was that if you take oncogene-driven cancer cell lines and you treat them with an appropriate matched targeted therapy, you often see evidence of ATM activation and gamma H2AX phosphorylation. So for example, these are PC9 EGFR mutant non-small cell lung cancer cell line. This is the HCC827, another EGFR mutant non-small cell lung cancer cell line. These cells are being treated with gefitinib. This was a first generation EGFR inhibitor, although similar results are observed with osimertinib. And what you can see is that even at pretty low doses and even on short time scales, we see very distinct activation of ATM and a DNA damage response. And that was interesting to us because historically, most of us who study targeted therapies don't think a lot about these drugs as directly driving DNA damage. But interestingly, we actually see the same trend in many other oncogene-driven models. So if you treat ALK-driven lung cancer cells with an ALK inhibitor, you see the same thing happening. If you treat BRAF mutant melanoma cells or FLT3 mutant AML cells with the appropriate targeted therapies, again, you see the same thing happen. And so we found this interesting, but I'll admit to you that when we first saw this result, we were very tempted to chalk it up to something trivial. And that trivial thing that we were tempted to chalk this result up to is the idea that, well, if these drugs are causing cell death, then maybe the DNA damage that we're seeing is just a consequence of that cell death. And if that's the case, that's not really interesting to us because we know that during apoptotic commitment, frank, full apoptotic commitment, cells can undergo DNA damage can occur. But the work started getting more interesting to us when we started realizing that the DNA damage responses that we're observing can really be fully uncoupled from cell death. So for example, if you treat cells at doses or over time intervals that do not yield any measurable changes in cell number, you still see evidence of gamma H2AX phosphorylation and ATM activation. Similarly, if you evolve cells to a state of resistance to the point where the drug to which they've been evolved resistance has no effect on proliferation, as is the case with these GR4 cells or WZR12 cells, these are EGFR mutant lung cancer cells with evolved resistance to EGFR inhibitors, again, you see the same phenomenon of ATM activation and gamma H2AX activation, again, despite the fact that the drugs aren't driving cell death. And so what this told us was that targeted therapies can drive cell death in a manner that's, can drive DNA damage in a manner that's uncoupled from cell death. We wanted to understand if indeed this was frank DNA damage that was causing this ATM activation. And so we used a COMET assay to indeed show that there was actual double-strand breaks that were being created. And we characterized the signaling downstream of ATM and showed that what we find is indeed, canonical double-strand break repair driven by ATM and CHEK2, whereas by contrast, we don't see evidence of ATR or CHEK1 activation, for example. And we see this in multiple cell lines. We started then asking, well, how is it that DNA damage might be occurring in these cells? And perhaps because of our background in apoptosis, one of the things that we looked at were markers of activation of the intrinsic mitochondrial pathway, and that work, that hypothesis was in part motivated for us by work from Stephen Tate and others in the apoptotic field in recent years, which have shown that cancer cells can undergo incomplete or partial mitochondrial outer membrane permeabilization, which is sufficient to drive what's known as sublethal caspase activation. So in other words, caspase enzymes can be activated even in cells that are surviving and growing. Indeed, what we found is that when we treat cells with these therapies at the same doses at which we start to see evidence of DNA damage occurring, we also see evidence of sublethal activation of intrinsic pathway caspases. If we further go in and actually knock out those key effector caspases of the intrinsic pathway, caspases 3 and 7, what we find is that fully reverses the DNA damage phenotype, and we can phenocopy that using QVD, pan-caspase inhibitor. And so that tells us that DNA double-strand breaks that we're observing in cells that are being treated with these sublethal doses of targeted therapies are being caused by sublethal caspase 3-7 activation. And finally, we leveraged some things that were already known about caspase-driven DNA damage to reveal that that it is the canonical caspase 3-7 substrate CAD or target CAD, which is responsible for this DNA double-strand break formation. So when we treat with targeted therapies, we see not only activation of caspase 3 and 7, but we also see degradation of ICAD. This is a protein that is an endogenous inhibitor of CAD, and CAD is a DNA nuclease that's known to drive double-strand break formation. So ICAD degradation would be indicative of CAD activation. And furthermore, we were able to prove that indeed it's this CAD activation that's responsible for the DNA damage phenotype. So if you knock out CAD, you block the ability of targeted therapies to drive DNA damage. Now, together, what this work suggested to us was a model which states that cells surviving targeted therapy nevertheless experience sublethal levels of caspase 3-7 activation. And this caspase 3-7 activation can lead to the degradation of ICAD, the consequent activation of CAD, and the formation of DNA double-strand breaks, and the consequent activation of ATM to drive repair of those double-strand breaks. Now, this model, once assembled, suggested something to us that we thought was pretty interesting. If you recall, I started this section of my talk by saying that residual cancer cells are really important to us because they are the seeds of eventual resistance. And of course, one of the defining features of residual cancer cells, in fact, the defining feature, is that they don't die when treated with therapy. Otherwise, they wouldn't be residual cancer cells. And so there's a great interest in figuring out strategies to target these cells And we reasoned that this pathway might give us such an opportunity, because indeed, if the cells that survive targeted therapy are undergoing this continual DNA damage process that requires ATM activation for its resolution, then perhaps inhibition of ATM could be particularly effective as a therapeutic strategy in residual tumors. And so we tested that hypothesis in a number of ways. First, we took different cancer cells, we treated them with sublethal doses of targeted therapies or ATM inhibitors alone, and we found that, as expected, that has no effect. Those have no effect. But when you combine those agents, you see a synthetic vulnerability. That's perhaps more clearly seen in this case of drug-tolerant persister cells. So these are cells that you can isolate by treating oncogene-driven cancer cell lines with lethal doses of targeted therapy and waiting for about a week and a half until you can identify and isolate the cells that survive. So these are the in vitro surrogate for residual disease. And in those cells, we find a similar thing, which is that targeted therapy does very little, of course, because these are, after all, drug-tolerant persister cells. ATM inhibitor alone does very little, but when you combine those two, you get a highly synergistic killing of these drug-tolerant persister cells. Again, likely reflecting back to this DNA damage process that's being driven by sublethal caspase activation. The idea that we can get rid of drug-tolerant persister cells by targeting ATM suggests that combining an ATM inhibitor with targeted therapies ought to reduce the size of that residual cell population, and as a consequence, make it much more difficult for resistance to eventually emerge. And we were able to show that in long-term time to progression assays performed in vitro, where, as you can see, cells treated with vehicle or an ATM inhibitor grow really nicely. Cells treated with targeted therapy experience a growth delay followed by the emergence of resistance, where drug-tolerant cells eventually grow out. But cells treated with the combination of targeted therapy and an ATM inhibitor never grow out despite long-term monitoring. It's worth noting that we see these same effects in many different cell lines. I've shown a lot of this work in PC9 cells, but we've seen this in, for example, in primary EGFR mutant lung cancer cells derived from patients. These were provided by Aaron Hata at MGH. We also see the same signaling in that setting. And we see that this result has clear in vivo implications, because if you form xenografts in mice, these happen to be different EGFR mutant lung cancer xenografts, and you treat them with osimertinib, which is the standard of care for these patients, what we find is very similar to what we saw in vitro, which is that, of course, vehicle or ATM inhibitor do very little to the growth of these tumors. EGFR inhibitor substantially suppresses the growth of these tumors before eventually giving rise to acquired resistance. But when you combine an ATM inhibitor with the EGFR inhibitor, you get a much more penetrant response, suggesting that you are more effectively eradicating residual tumor cells, but you also get a more durable response. And so this is read out in tumor growth curves, but also in the number of individual tumors that grow out past a threshold size as a function of time, which you can see on these plots on the right. I will point out that all of these results, that is the sensitization of residual cells to ATM inhibitors, these are all dependent upon the activation of caspase 3/7 and CAD in these tumors. So I haven't shown it here just for the sake of brevity, but these effects are all caspase 3/7 and CAD dependent. And lastly, I just want to conclude by saying that that comparable or analogous effects are observed in human cancer patients. So, for example, we teamed up with Trevor Bavona and Colin Blakely and a number of their colleagues at UCSF to look at tumors at the treatment-naive or residual disease state. And what we observed is that in the treatment-naive state, we see low levels of ATM activation. These are, by the way, are all EGFR mutant lung cancer tumors treated with EGFR inhibitors. What we see, by contrast, in the residual state is a marked upregulation of ATM. So consistent with this idea that in surviving tumor cells, you're getting upregulation of this DNA damage response. Furthermore, we see that by working together with Helena Yu at Sloan Kettering and the Sloan Kettering genomic sequencing database, what we were able to see is that in rare EGFR mutant lung cancer patients, who have loss of function mutations in ATM, they respond considerably better to upfront treatment with EGFR inhibitors than patients who are ATM wild type. It's important to note that this study, this correlative study, involves only a small number of patients because ATM loss of function mutations are quite rare in EGFR mutant lung cancers. But nevertheless, there's an interesting signal of efficacy there. So with that, I'm just going to conclude and thank you for your attention and just summarize the work that I described by saying that, in the first part of the talk, I attempted to describe how we have tried to build on our foundational understanding of the mechanisms of BCL2 interactions in the mitochondria and how those regulate apoptotic commitment, and really add to that understanding the beginning pieces of a broader understanding of how other broader features of cancer cells impact the regulation of this important network and exploiting that information for potential clinical benefit. And then the second part of the talk, I described our work on residual disease and how we've uncovered the phenomenon of sublethal caspase-3/7 activation and consequent ATM dependence in residual tumor cells that can be exploited to minimize or eradicate residual tumor cell populations. So with that, I want to thank you for your attention. I do want to acknowledge the folks here pictured, which are the current members of my lab, the collaborators who worked with us on the studies that I described today, and our funding sources. And with that, what I'll do is exit presentation mode, but continue to screen share so that I can see the Zoom screen and take any questions. So thank you.
Great. Thanks for that, Chris. It was an awesome talk. Great overview of your work thus far and the latest things that you all have been up to. We have a few questions coming in from the participants. Just a reminder to everybody, if you have a question, you could submit it into the Q&A box at the bottom of the screen, and I'll be happy to communicate those to Chris. So if you have a few minutes, I'll ask the first one. This is from Iram Khan. The question is, does the type of cancer treatment affect resistance mechanisms? Would you predict the same resistance mechanism applies to cytotoxic chemotherapy, radiation, and epigenetic therapy?
Yeah, it's a good question. I wish it were that simple. Unfortunately, I don't think it is. I think that the resistance mechanisms that occur are very much templated by the selection, the details of the selection pressure that's applied. And so as a result, the resistance profile that we see is very much a function of the particular drug that we use.
Okay, and then another question came from Zintis Indy. Given their distinct mechanisms of action, why do both BRD4 and BRAF inhibitors drive activation of MYC signaling in resistant cells? And what other drug classes, if any, do you think might similarly converge on MYC?
Yeah, I mean, this is a great question, and I'm not sure that I have a great answer to it. So in the setting of AML, prior work, as many people in the audience probably know, has implicated MYC suppression as being critical to the mechanism of efficacy of BRD4 inhibitors. And so it's not surprising that in that setting, that the rescue of MYC suppression might be something that would be observed in the setting of resistance. In melanoma, the role of MYC was a bit more surprising. I think that initially we didn't necessarily expect MYC to be a critical part of the resistance paradigm, but our studies pointed us to MYC, and ultimately we were able to reconcile that finding based on the fact that if you look at the key resistance mechanisms that have been shown in melanoma, nearly all of them, if not all of them, are canonical activators of MYC. And so, we think that it could just be that MYC is one downstream thing that can rescue the toxicity of these drugs, and so it's something that evolution finds over and over again.
Interesting. Next question is from Daiqing Liao. He or she asks, how does DNA damage response via ATM confer survival?
Can you repeat the question at the end?
So how does DNA damage response, which I'm assuming is meant by DDR, via ATM confer survival? So I guess the question is, how does ATM activation help the cells survive that sublethal caspase activation?
Yeah, great question. We don't know the answer to that. I mean, obviously, ATM can activate a number of key effector pathways. This is one of the things that we're working on right now is to really try to resolve what are the key effector pathways downstream of ATM activation that are really rescuing survival in these cells. I think stay tuned.
All right, and then the next question from William Beck. Thanks for the nice talk. The MYC story is interesting. MYC controls expression of the splicing factor PTB-P1, which in turn controls splicing of PKM between PKM1 and PKM2, the tumor form driving aerobic glycolysis. Are you seeing alterations in O2 and other metabolic functions?
Yeah, it's interesting. We definitely see changes in metabolic state in resistant tumors that are MYC dependent. We definitely see changes in sensitivity to different types of drugs that impinge on metabolic pathways. We have not attempted to link that to PKM1, PKM2, which would, I think, be an interesting mechanistic link if it were true. But it's unquestionably true. And that's also, I think, squares nicely with work by a number of labs that have implicated MYC-dependent metabolic changes and metabolic dependencies in MYC-driven tumors.
Okay, another question came from John Abrams. Conventional wisdom suggests that caspase-3, caspase-7 activation is cleaning up the corpse, and the point of no return is loss of cytochrome C in mitochondria. Can you comment on how your observations on CAD, ICAD resolve with this? Also, have you looked at CAD, ICAD in single cell studies?
Yeah, really great questions. The easy answer to the second question is no. We haven't looked at single cell resolution. That's very important. The answer to the first part of the question, I think, is that I think most of our work suggests that that mitochondrial outer membrane permeabilization is really important for is a point of no return with respect to cell death induced by a number of targeted therapies. It is not clear, though, that caspase activity is actually required to execute that cell death. And in fact, so you can, for example, apply caspase inhibitors to tumor cells that are driven by different oncogenes, and simultaneously treat those cells with oncogene targeted therapies. And the caspase inhibitors won't block the cell death that is caused by the targeted therapy. They might change the nature of the cell death that occurs, but caspase 3 and 7 I don't think are going to be required for that. So I think MOMP is a point of no return. I'm not sure that the caspases are essential for it.
And it probably depends on whether it's minority MOMP or sub-maximal MOMP versus the complete MOMP that sometimes we refer to as that point of no return. So
sure, absolutely, great point. That's right. That's right.
It's becoming more of a gray area than it ever has before.
That's right.
Another question, Chris, is you kind of paint this narrative that we can potentially take advantage of BH3 mimetics by forcing cells into a more dependent state. So regardless of what we start out with, we can give different kinds of therapies, drive them into a sensitive, vulnerable state for those BH3 mimetics, and then execute with those agents given at the proper time. So beyond heme malignancies and the couple of examples that you gave us nicely here, where else do you think there's real opportunity for doing such a strategy? So coming in with some kind of agents specifically to try to drive these dependencies. So what cancers do you feel like are most amenable to such a strategy, and which drugs do you feel like would work well in that setting?
Yeah, great. It's a great question, Chris. And I think for those of us who have looked at how targeted therapies influence the BCL2 network, we all appreciate that in many different contexts, the treatment of cancer cells with particularly targeted therapies yields a dependency, a heightened dependency on BCL2, MCL1, BCL-XL, or combinations thereof. And that seems to be something that shows up across many different solid tumors, KRAS mutant tumors, PI3 kinase mutant tumors, EGFR mutant tumors. And so while indeed you're correct in saying that venetoclax, the BCL2 inhibitor that's of course obviously the most advanced BH3 mimetic drug in the clinic right now, has largely shown activity in heme malignancies, I think that the future very well may hold many, many examples of cases where the activity of therapies applied to solid tumors, particularly targeted therapies, will be improved by combining them with BH3 mimetics targeting things like MCL1 and BCL-XL. It turns out that BCL2 is probably not as effective in solid tumors in that way. But that's going to be really dependent upon is, can we get MCL1 or BCL-XL inhibitors that can be applied safely in patients? I think there's been remarkable work in the last few years that gives a lot of us, I think, in the field excitement that might very well be the case. But I think really we're going to find out more in the next few years.
Yeah, I definitely agree with you there. Something else that we're very well aware of in the field of cell death is the emerging data showing that mitochondrial apoptosis can be much more inflammatory in terms of the signaling that emanates from dying cells, given the release of mitochondrial DNA from these back-to-back macropores, and then in the absence of caspase activation. So do you think that there's some opportunities to go that route and also be able to enhance the sensitivity of cancers to immunotherapies and strategy like that? And where do you see the studies that your lab has done as fitting within that type of treatment paradigm?
Yeah. This is one of the most fascinating areas in the cell death field, I think, right now, at least as it pertains to cancer cell death, is this phenomenon that, when you permeabilize the mitochondria and you activate caspases, you are potentially doing a lot of things to a cell that could, in the right setting, be stimulate inflammatory signaling. And furthermore, I think there's some really compelling evidence out there that caspases, among other things, might be restraining some of that. And so increasingly, I think a lot of folks are understanding apoptosis to be a means of dying without causing inflammation, and that might be an important distinctive feature of what really apoptotic cell death is all about. And so by understanding the underlying mechanisms, as you're suggesting, I do think that there's a chance to use targeted agents to induce or further hyperstimulate inflammatory signaling from tumors in the context of these combinations. And that's certainly stuff that we and others, I think, are working on now.
And so would you think that the combinations that you're utilizing with ATM inhibitors could be leveraged in such a manner, perhaps with a caspase inhibitor or some other maybe APAF inhibitor at the same time,
right?
To then take advantage of that?
That's right. I think that would make a lot of sense, right? If indeed, this ATM signaling or caspase signaling are contributing to inflammation or have the potential to drive inflammation, then modulating those therapeutically should accentuate that. Now, I don't want to oversimplify what a tumor immune response requires. There's a lot that goes into that, and I think we need to understand with more resolution really what are the tumor types in which this type of manipulation, which drives interferon signaling, for example, would be enough to get you over the hump and really confer sensitivity to something like a checkpoint blockade, and what tumor types that wouldn't quite be enough because of neoantigens and the like. So I think there's a lot to be resolved, but I think what's really exciting is the more we appreciate that apoptotic cell death is not just about cells dying, it's also about inflammation. And connect that with what's already known with respect to inflammation and anti-tumor immunity, I think we're laying the groundwork for some really exciting opportunities to really combine molecularly targeted therapies with immune therapies using this understanding.
Great. We'll have time for one more question. So this one comes from Nama Kanarek. And she asks, do you predict that ATM inhibitors will be effective only after the first round of chemo is given, or will it make sense to treat with sublethal chemo plus ATM inhibitors simultaneously?
Yeah, it's a great question. Hi, Nama, by the way. Yeah, I think that either one could make sense. You could imagine treating with a single agent targeted therapy until you reach a minimal disease state, and then adding an ATM inhibitor on top at that point. You could also imagine just coming in with both at the same time. We are trying to start a trial right now, and I think what we're going to try to do is just lead in with both agents together. but I think both could make sense. Because, the targeted therapy on its own as a single agent is, based on everything we know, sufficient to kill many of the cancer cells to begin with. So you can clear those out with just the targeted therapy alone. I think it's those cells that survive that we need a solution for. And ATM signaling and ATM dependence might be at least part of the solution there.
Very cool. All right, Chris, that's all the time that we have today, but we appreciate you coming so much today and chatting with us, sharing your latest data. It was a fascinating talk. Thank you so much for participating, and we look forward to seeing you again soon.
Great. Thanks so much. Bye-bye.
Take care.